Number 26. In this podcast, we are going to talk about protein structure, and then later on this week, we'll talk about how structure can affect function. Right in our roadmap here, again, this book skips around a bunch. Chapter 7, we're on cells. We'll talk about cells again in 8 and 9 this week. Okay. So there are 4 levels of protein structure that affect how they work. They affect function. They're called primary, secondary, tertiary, and quaternary. Primary, secondary, tertiary, and quaternary. All of these are pretty easy to understand except for secondary, and I'm going to give you a little tip on how to do that. So primary structure is just the order of amino acids. So this is the 1-letter code for amino acids. You don't have to memorize this, but this stands for methionine, phenylalanine, glycine, arginine, tyrosine, threonine, tryptophan, so on. So each one of these letters is an amino acid, and just the order of amino acids that go into making a protein is its primary structure. Just the order of amino acid. Amino acid number 1 is methionine. The last amino acid is leucine. And all the amino acids in this order in between, that's the primary structure for a particular protein. And there's a peptide bond between all of these. This is the order in which the ribosome put them together, reading the codons from the mRNA. The secondary structure is the one that people have trouble with, and this is local folding within a chain. So if this is the first amino acid here in this chain, and this is the last amino acid here over in that chain, as the protein is made, it'll start to fold locally within that chain, not the whole thing folding up, but just locally within that chain, and you might get what's called a helix or a beta-pleated sheet or something like that. That's an example of secondary structure. Now that's the thing that is that people have the most trouble with when talking about protein structure, but I'll go over this again and hopefully you'll understand it when I use an analogy. Okay, there's an alpha helix. And these are beta strands. Not a great picture. This is a hand-drawn picture, I think, by one of the authors. But the symbols in protein structure lingo is they use arrows for secondary structure. So arrows like this for a sheet, and then a curled up helix for a helix. And these are stable, and the reason that we talk about secondary structures is you see them over and over again in all kinds of different proteins, and they confer different kinds of properties on those proteins. And so it's useful to talk about secondary structure because it's a feature, something that you will see on lots of different kinds of proteins. And tertiary structure is just one chain all folded into a 3D shape. So if we go back here to our chain, and if we take this chain and we fold it up into its 3D shape, its final 3D shape, that's called tertiary structure. And all proteins have primary, secondary, and tertiary structure. Primary, secondary, tertiary structure, okay? Because all proteins have at least one chain. And some proteins have more than one chain. So those proteins have quaternary structure. In that here's 2 different chains folded up, one in yellow, one in purple. 3 chains folded up, one in red, one in purple, one in yellow. Each one of these has its own primary, secondary, and tertiary structure, and then all 3 of them together form a quaternary structure. So this is not considered 3 proteins that work together. This is considered one protein that has 3 parts. There would be 3 different mRNAs that are code for this protein. They would each translate a different chain on the ribosomes, and then after all those 3 chains are translated, then they come together to form the functional proteins. Not all of cells' proteins have quaternary structure. Some stop at tertiary structures, just one chain folded up, and then it goes and does its job. But many have many chains, and some are very complicated. So we'll talk about, next week, we'll talk about how you make energy from breaking down glucose. There's a protein at the end of that biochemical pathway called cytochrome oxidase. And that protein has, instead of 3 subunits, it has 13. So 13 different chains have to come together and fold into the correct shape. 3 of those are coded for by the mitochondrial genome, and then the other 10 are coded for by the nuclear genome. So the nucleus and the mitochondria have to coordinate when they're going to transcribe those genes. Then each one has to be translated separately, and then they all have to come together to form this big 13-unit protein. And if that doesn't work, you die, because you need that protein in order to live. So assembly of proteins can be really complicated. Again, not everybody has quaternary structure. Not all proteins have quaternary structure, but many do. So one way to think about secondary structure, so if you think of just a human outline, and it folds up into this 3D shape that looks like this person. Local structure within this 3D shape would be secondary structure. So for example, I can recognize an arm here. I can recognize a foot here. And in fact, if I looked at another protein, I might also be able to recognize something that will look like an arm or something that looked like a foot. So that's secondary structure, is local folding within the one chain that's going to fold up and form one 3D shape. I can recognize things within that 3D shape, like an arm or a foot. The protein version of that would be like an alpha helix or a beta pleated sheet. So that's an example of secondary structure. Quaternary structure would be many tertiary structures working together to form one unit. So if this is a soccer team, one soccer team plays another soccer team in the game of soccer. It's not that one individual plays against another individual in soccer. It's a team that plays against another team. And that's how some sports work. Some sports, individuals play against each other, like singles tennis. One individual would play against another individual, and that would be like 2 proteins that had tertiary structure. Whereas the protein unit has multiple parts to it, made up of multiple individual chains. That would be an example of quaternary. And then if it had one chain all folded up, and that was how the protein worked, that would be an example of tertiary. So once again, all proteins have primary, secondary, tertiary structure. Not all proteins have quaternary structure. Okay. So how is it that a protein comes to fold into a particular shape? It all depends on the order of those amino acids and the property of their side groups. So the side groups here are called the R group sometimes, and they are indicated in this gray box in each of these. And they're characterized by how much they like or don't like water, and also some other properties. Now, in your book, it says that these 9 amino acids have hydrophobic side groups. It means that this gray box in the side group does not like water, okay? And it's hydrophobic. And so when these proteins are folding in a chain, these are going to fold so that they are getting away from water. So they tend to be toward the inside of a protein if water is on the outside of the protein. Okay. Um, there are other side groups, this time in a white box, that are hydrophilic. They like water. So they will tend to go toward the outside of a protein. There are also charged amino acids, okay, that they have. These have a positive charge on the outside of them. These have a negative charge on the outside of them. Positive charged amino acids and negative charged amino acids tend to stick together in a protein. So if you have amino acid number 3, that's an arginine, and you have amino acid 222, that's a glutamic acid, they will tend to bend the protein together so that those two side groups can interact. Now, having said this, okay, it's not important that you memorize who's hydrophobic and who's hydrophilic and who's charged. In fact, if you look at 10 different intro bio books or even biochem books, they will place these in slightly different groups. Sometimes glycine is considered non-charged. Sometimes it is considered hydrophilic. It just depends on the rules that the authors are using. Okay, it's more important that you know that for most amino acids they have this common structure. Okay, you can see this is common to all of them. Okay, and how they differ is by this side group. And these side groups have different characteristics. They can be hydrophilic, they can be hydrophobic, they can be charged. Now they're cheating a little bit with this one, proline, because if you notice, it doesn't quite have the same structure here as all the others. When proline binds to another amino acid, it tends to put a big kink in the chain. So when there's a mutation where there's normally a proline there and you change it away from, the mutation changes away from proline to something else, or the opposite, when it's normally something else and you change it to a proline, that can really affect the function of the protein because of how the chain folds. And protein or proline usually puts a big kink in the chain. So if you take that kink out or you put an extra kink in, that changes how the proteins fold. Okay? So as the ribosome is putting together these amino acids, they all have different characteristics. Some like water, some don't like water, some like each other, like positively charged and negatively charged, and that affects how they fold. Now, your book doesn't really talk about this, but in other classes you will find out that most proteins don't just fold spontaneously when they come out of the ribosome. They need help arriving at their final three-dimensional structure, the structure that is needed for the protein to do its job. Okay? If you think about something like your car seat does not work like a car seat if it's shaped like a sword, for example. It needs to be shaped like a car seat in order for to do its job as a car seat. So proteins need to have the right shape in order to do their job, and they don't spontaneously form into that shape most of the time. So how do they do that? There are other proteins that help them fold into the right shape. Those are called heat shock proteins or chaperones. And they bind protein chains as they're coming off the ribosome and then help them fold into the right shape. Okay. Once a protein has achieved its final shape, it's not a rigid shape. It's flexible, and it's got to be able to flex in order to do its job. If you think about something like the shape of your hand, okay, your hand needs to have a hand shape to do its job, but it's not completely rigid. When you shake somebody else's hand, and hopefully this is somebody within your household so you can maintain good social distancing, your hand changes shape and their hand changes shape. It's not rigid. It's not like you're shaking a block of granite. It changes shape. Okay? So we're going to talk about different ways that proteins can be change their shape and it affects their function. And those two different ways are allosteric and covalent. Okay. There are energy molecules in the form of carbohydrate that can be stored in food. This is why we eat food. So when we eat food, we can take apart those bonds in that food and convert that energy into a form that we can easily use. And we'll talk more about that next week. Some form of how those bonds are put together is easy for humans to convert from one form to another, like starch in a potato, and some is not. So starch has a connection between these glucose molecules. You're going to see a lot of these next week. Okay, here's a glucose monomer, one unit of a glucose molecule linked together with another unit of a molecule, glucose molecule, and you can link a whole bunch of these together in a form called starch. And it has a 1,4 glucose link between starch units. So carbon number 1 on one unit is hooked up to carbon number 4 on another unit. So this carbon number 1 would be hooked up to another glucose unit, carbon number 4, and so on, over and over and over again. Okay? There is another molecule that is made up essentially of the same stuff, the same atoms as is starch, and but they're hooked up differently, and we call that chain of glucose units a different name, cellulose. Cellulose is what makes the cell wall of plants. It is essentially wood is mostly cellulose. So if you are crunching through celery and you feel like those hard bits, stringy bits in celery, you cannot digest those. So you cannot take apart those bonds and use them for food. And so they are indigestible and they help you poop. And what makes them indigestible is between carbon number 1 and carbon number 4, there's a beta bond. It's a different shaped bond than what you saw between carbon number 1 and carbon number 4 in starch. This is an alpha bond. Okay, you see how these two glucose molecules are lined up right next to each other in a plane or a flat line, whereas here they're tilted across each other in a beta bond. And the proteins that we have that recognize this shape proteins that we have that recognize this shape, okay, this shape of bond, and it's going to break these apart so we can get at these glucose molecules. They don't recognize this shape. So even though the glucose molecule is the same, they can't recognize this shape here, and so they can't break that down, and that's why you don't digest it and you poop it out. That's not saying that that's the only thing in the plant. Obviously, you eat celery. It's delicious, and it has glucose molecules in there, and it has some starch molecules in there too. But the cell wall body of the plant is made up of cellulose, and you don't digest that part. Okay? So the bond angle is critical. Your proteins recognize this, don't recognize that. Okay? So these are different covalent bonds. Termites can break the beta 1,4, and it's actually not the termite that breaks that bond. It's bacteria that live in the gut of the termite that break that bond. And they harbor those bacteria in their gut so that they can chew up the wood. The bacteria digest, break this bond, and then the termite gets the access to these sugars. We don't harbor those bacteria in our gut, and so therefore anything with this kind of bond, we can't get at those glucose molecules. But if it has this kind of bond, we can. So this is an example of covalent modulation of protein function, in that you have a certain shape, it can bind to one thing, it can't bind to another thing. And the enzyme that breaks down this bond can recognize this shape, but it can't recognize that shape. Okay? In the next podcast, we'll talk about allosteric regulation, which is a different sort of regulation of protein function.